Okay, good evening, students. Hope I am clear. My voice is yes. Thank you. So, you have learned about nerve physiology in your intermediate thoroughly while you are preparing for NEET examination. I think so. Even in your long term, you might have heard about it. And in degree, you have studied in your third semester. And now, I'm daring to teach you what you have already learned previously. So, I'll be cautious to make you understand this entire concept. There are just 5 or 6 bits that you have to uh, take into consideration in this topic, that is origin and propagation of nerve impulse, for which you need to have the complete understanding of the physiology of the neuron. Unless otherwise, you cannot attempt those questions if they are said to be somewhat complicated. Okay. If it is a direct one word sentence questions, you can answer it. If it is somewhat related to the process and one after the other is depolarization first, sodium potassium pump first, or repolarization first, hyperpolarization. So the sequence of events also sometimes they may give. So you please try to understand this. If you have any doubt, you can ask me at any time. Okay. So coming to the nerve physiology, I shall start with neuron. Okay, neuron is the structural and functional unit of nervous system. We all know that. It is also considered as the longest cell of the body. It is also called as the longest cell of the body. And you have studied the structure of the neurons so many times, and let me explain this once again. We can study the structure of neuron with respect to its nerve fiber. So based on the nerve fiber, or which is also nothing but the axon. Neurons are of two types. Okay, and one is said to be myelinated nerve fiber, neuron with myelinated nerve fiber, and a neuron with non-myelinated nerve fiber. And in another one, neurons are of different types based on the processes that arise from the neuron, like unipolar neurons, where there is a cell body and only axon. Bipolar neurons, where there are dendrites on one side and axon on the other side of the cytone. Multipolar neurons, where there are dendrites on different sides of the cytone and an axon on one side. And there is a pseudo unipolar neuron. The name it implies that it is actually a bipolar neuron. But what happened is this part, the cytone, moved to one side so that the axon and the dendrites are present on the other side. So a bipolar neuron with dendrites and axons on one side is called as a pseudo unipolar neuron. Thus, we have four different kinds of neurons. One is unipolar neuron, where there is an axon with cytone. A bipolar neuron with dendrites on one side and axon on the other side. Multipolar neuron with a number of dendrites on different sides of the cytone and an axon on the other side. and a pseudo unipolar neuron, which actually during the embryonic development is a bipolar neuron that modified to form a unipolar neuron. So in all these four types of neurons, we come across with commonly three structures. One is called as the cyton. So the cyton is nothing but the cell body. It is the cell body. Cell body means it has a plasma membrane and it has a nucleus, it has cytoplasm and all other cell organelles that are characteristic of a eukaryotic cell. In addition to this, it has number of processes. I am drawing here a multipolar neuron, and these processes are called as dendrites. Okay. In addition to this, scientists have observed certain granular structures, and the scientists who observed these granular structures are called as is nothing but Nissl, based on which based on his name these are called as Nissl granule. Whenever you come across with the term granule, okay, and this term granule is one which is a vesicle that comprises of certain substances inside, stored substances, or which can be stained with a certain dye so that under the microscope it appears as a granule. And here the scientists say these Nissl granules are not these vesicles that generally we speak of. Here they are called as the ribonucleoprotein particles. Here they refer to them as the ribonucleoprotein particles. And on the other side of the cyton, there is a portion from which the axon arises, and this is called as axon hillock. And from the axon hillock, the axon continues, and this is nothing but the axon or nerve fiber. So axon is also called as the nerve fiber. The nerve fiber at the other end divides to form a number of telodendrites. Telodendrites. Each telodendrite, the branches of the axon, end up in knob-like structures, and these are called as synaptic knobs or terminal buttons. Terminal buttons or Boughton terminox, whatever it may be. Reversing these two, these are Boughton terminox. So they are present at the termini, ends of the axon, and they are found to be like a button like structure, hence they call them as Boughton terminox. In addition to this, in some neurons, the axons show certain lateral processes. These lateral processes are called as collateral fibers. The lateral processes of the axon are called as collateral fibers, and these are responsible for forming junctions with the adjacent neurons. Okay, this is the overall structure of the neuron. And when such neurons are present side by side, there are several neurons present side by side in a nervous tissue. Like muscle tissue, this is also having three levels of connective tissue surrounding it. Around the nerve fiber, that means there is a connective tissue called as endoneurium. Around each neuron, there is a epineurium. Around the entire nervous tissue, there is a perineurium. So these are the connective tissue layers present around the neuron. And removing these connective tissue layers, we come across with this structure in the nervous tissue, where a number of neurons form into a nervous tissue. In this nervous tissue, we can observe different nerve fibers are present side by side, and all the cytons are present side by side. So when you see it under a microscope, it appears as a lump here like this, and all the nerve fibers appear like this as a strand. Okay, and this we call it as nerve. What is nerve? It is nothing but a group of nerve fibers, that means axons, in a nervous tissue, and this is called as a ganglion, which is nothing but a group of cytons in a nervous tissue. Okay, and these two terms are associated with peripheral nervous system. When the same nervous tissue present in the central nervous system, brain and spinal cord, instead of ganglion, we use the term nucleus, which I have explained already while explaining the pituitary gland. There are two nuclei which are responsible for the production of neurosecretory hormones that are stored in the pars nervosa, respectively vasopressin and oxytocin. And this is called as supraoptic nucleus and paraventricular nucleus. So thus nucleus is nothing but ganglion or group of cytons present in central nervous system. And the same name, the nerve in central nervous system, is called as a tract. So these are the terms which you have to get familiarized with. So nucleus and ganglion are same, and they refer to the group of cytons only. Ganglion in the peripheral nervous system and nucleus in the central nervous system. Likewise, group of nerve fibers are having two names. One that is nerve in the peripheral nervous system, and the other that is tract in the central nervous system. Okay, so this is the basic structure of a neuron, and based on the nerve fiber, neurons are again of two types. I have said before: neurons with myelinated nerve fiber and neurons with non-myelinated nerve fiber. So this is a neuron, and this neuron is having an axon, and this axon is not surrounded by any other membrane. This is simply axolemma. Okay, the membrane surrounding the axon is called as axolemma, and the membrane surrounding the cyton and dendrites is simply called as the plasma membrane or cell membrane. So that means the plasma membrane of the axon is called as axolemma, and this is the cytoplasm present in the cyton, and the same cytoplasm present in axon can be called as exoplasm. So apart from these, there is no much what we call difference in the structure that we have just uh, discussed now. Now coming to the myelinated nerve fiber. So in a myelinated nerve fiber, what happens is, so there is a cyton followed by axon hillock and neuron followed by telodendrites and Boughton terminox. And here there are specialized cells associated with the neuron, and these cells are called as, named after the scientists who discovered it. In the morning class we have listened to the cell theory. They say it is proposed by Schleiden and Schwann, and here this Schwann is said to be recognized, identified these cells, hence named after him. These are called as Schwann cells. These are called as Schwann cells. Just a second, I get back to you. రాజు,
సార్
అర్జెంటుగా
చెప్పండి సార్.
బయోటెక్నాలజీ దాని మీద ఉంది కదా ఆ పైన, దాని సైజు ఉందన్నావు కదా, మనోడికి చెప్పి
సార్.
మీడియా సెంటర్ అని మాత్రమే వేయించు. పైన ఒక చిన్న లైన్ డాక్టర్ విఎస్ కృష్ణ గవర్నమెంట్ డిగ్రీ కాలేజ్ అటానమస్ రావాలి, దాని కింద మీడియా సెంటర్ పెద్దగా రావాలి. ఓకేనా?
ఓకే సార్.
రెండు వైపులా, ఇటుకి అటుకి కూడా. రెండోది, నీ బ్యానర్ సేమ్ నువ్వు ఏదైతే తయారు చేసావో దాన్నే 2x3 22 సైజులు అనుకున్నావు కదా, 2 సైజులు వేయించి. రెండు సైజులు. ఓకేనా?
రేపు మార్నింగ్ సార్.
రేపు మార్నింగ్ వచ్చేయాలి. 10 o'clock మనం ఫిక్స్ చేసేయాలి. నేను 9 కల్లా కాలేజీలో ఉంటాను. నువ్వు నైటే వేయించుకుని వచ్చేయి. ఓకే, బాయ్. Sorry for the inconvenience. So here, these Schwann cells are present. The Schwann cells secrete what we call a concentric layers of lipids around them. So this concentric layer of lipids are found wherever we come across with the Schwann cell. Okay, and this concentric layer of lipids is called as Schwann sheath or myelin sheath. Hence, these nerve fibers which are surrounded by a myelin sheath are called as myelinated nerve fibers, and they are also called as medullated nerve fibers. Here the medulla is the term used by some scientists for the myelin. So they can be called as myelinated nerve fibers or medullated nerve fibers. And the one without this myelin sheath is called as non-myelinated nerve fiber or non-medullated nerve fiber. And what is the significance of this myelin sheath? Okay, the myelin sheath significance is that it is made up of lipids. It is mainly made up of lipids. Lipids are non-excitable. Lipids are non-excitable. That means the region where this particular myelin sheath is said to be present, whenever you give a stimulus here, the nerve is not going to be excited. Then what happens where the actual excitation takes place? It takes place between two what we call myelin sheaths. So the region where the myelin sheath is present is called by Ranvier as node of Ranvier, and the region between these two nodes is called as the internode, and where the myelin sheath is absent, that is the region where it is actually excitable. It is actually excitable. And what is the significance? And whenever here we come across with this nerve fiber, myelinated nerve fiber, so excitability is restricted to this, so that the electricity that is generated passes from one node to the, this is the node, from one node to the other node, and this place is called as internode. Okay, it passes from one node to the other node. That means this much distance is said to be jumped, so that it travels at a faster rate. So for faster conduction, wherever in the body faster conduction is needed, there the non-myelinated nerve fibers are present. Okay, and coming to the neuron has certain properties. The first property that we have just heard is excitability. So a neuron or a muscle, both are excitable. Given a stimulus, they can be excited, but they should be given a stimulus. And what kind of stimulus they have to be given? The stimulus that they get excited is called as a threshold stimulus. Okay, what is a threshold stimulus? It is the minimum stimulus that is required to excite a neuron or even a muscle is called as threshold stimulus. The minimum stimulus that is required to excite a neuron or a muscle is called as threshold stimulus. Let us say there is a group of neurons, okay, and this can be excited at about 5 millivolts of what we call energy, electrical energy, or whatever, one kind of stimulus. If you give only 4.9 millivolts or even 4.95, it will not excite. It will get excited only this minimum stimulus is reached, and hence we call this stimulus minimum stimulus. That it means even if you give 6 millivolts, it responds. 7 millivolts, it responds. But what is the minimum stimulus that is required? 5 millivolts. If it is below 5, it will not respond. That is what a minimum stimulus or threshold stimulus is. So based on this, there is a law that is proposed that is called as all or none law. All or none law. What does it mean? Here, until the threshold stimulus is reached, the neuron will not respond at all. That means there is no response until the threshold stimulus is reached. Once the threshold stimulus is reached, the neuron responds totally. Thus, it is all. And based on these two, we call this as an all or none law. In addition to this, a neuron or a muscle has another characteristic, that is what we call it as conduction. So excitation is not sufficient. Once it gets excited, the excitation has to be conducted all along the path, all along the length of the neuron, from dendrites to telodendrites or from telodendrites to dendrites. And in addition to this, this exhibits another phenomenon called as refractory period. Refractory period, which I will explain when we deal with muscle. Not to waste our time here, I will explain it there. And coming to the actual function of neuron, that is nerve impulse. Okay, transmission of nerve impulse. So this can be studied under two heads. One is called as origin of nerve impulse, the other is called as propagation of nerve impulse. So, what is the definition of a nerve impulse? Scientists say it is the sum total of physical and chemical events that are involved in the propagation of a wave of physiological activity along a nerve fiber is called as nerve impulse. The sum total of physical and chemical events that are involved in the propagation of a wave of physiological activity along a nerve fiber is called as nerve impulse. So here, as the definition says, here it is a wave. It is nothing but a wave of physiological activity that is propagated. And how this wave of physiological activity is generated? It is generated by certain physical and chemical events. So before I go for the origin and what we call uh, propagation of nerve impulse, let me explain the properties of cell membrane. So the cell membrane, in the morning you have heard that it is said to be having a property called as permeability. So permeability is of two types: semi-permeable membrane and selectively permeable membrane. So the plasma membrane can be either semi-permeable or selectively permeable. No biological membrane is impermeable. Okay. In generally, maybe there may be exceptions that I have not studied maybe, but actually what we say is membranes are biological membranes are not generally impermeable. They are either semi-permeable or selectively permeable. So if this is the plasma membrane, substances have to be transported into and out of the plasma membrane. They have to be transported into and out of the plasma membrane. And for this reason, okay, the only hindrance is the plasma membrane with lipids as well as proteins. According to fluid mosaic model, the lipids are said to be arranged in bilayer with proteins that are called as integral proteins, some of which act as carriers, and some of them are said to be acting as channels, and some of them are said to be acting as carriers only with respect to the transport of substances against concentration gradient. So there are two types of carriers. One carrier does not use any energy at all, that is involved in passive transport. Another carrier which acts as an enzyme breaks down ATP, releases energy, and that is involved in active transport. And another protein present in the plasma membrane acts as a channel. So all these carrier proteins have specific binding sites for the ligand or the substance to be transported. So everything has a binding site based on which either the channel opens or closes. Okay, so here there are two. Uh, Whenever we come across with transport of substances, transport of substances takes place in solution, isn't it? Solution has either solvent or solution has a solvent and a solute. A semi-permeable membrane is the one which allows the solvent but not solute. What kind of solute? It will not allow the solute which is said to be somewhat larger in size. So this allows solvent but not the solute. Hence it is half permeable, semi-permeable membrane. And coming to the other one, selectively permeable. It allows the solvent to pass through and allows the solute also to pass through. But the only difference is, let us say there are three kinds of solutes, A, B, C, D. Okay, three kinds only, A, B, C. So here there is a biological membrane. It will allow A, will allow B, but not C. That means it has selected only E and B, but not C. So based on what? Based on its physiological activity. For example, excretion. Excretion involves elimination of urea. So, whenever there is a selective reabsorption in the uriniferous tubule, glucose gets reabsorbed, bicarbonates get reabsorbed, sodium gets reabsorbed, everything but not urea, because it is the physiological function attributed to the excretory system. Thus, it is selectively permeable. So, thus, there are semi-permeable membranes and selectively permeable membranes for which we come across with a number of proteins in the form of carriers or channels. So here, coming to the plasma membrane, where there are channels. Okay, so here there is a channel, and this channel is said to be having a gate-like extension which closes it. And I've already told you that it is having a binding site for the ligand. And each channel is always specific. Channels are specific for a particular ion. For example, if you say it is a sodium channel, it allows only sodium ions. If you say it is potassium channel, it allows only potassium channels. And in all biological membrane, what the scientists have found is every biological membrane, when compared with sodium and potassium, there are more number of potassium channels than sodium channels. Thus, you study when you study like this. Potassium has more permeability than sodium ion. What does it mean? How come it is possible? Only because of the fact that there are more potassium channels and less sodium channels. And inference we say potassium is more permeable than sodium ions. Then if it is a sodium channel, it has to open only when the sodium ions come there. How it takes place? The sodium ion binds to the binding site. Whenever the sodium ion binds to the binding site, immediately the channel opens. The channel opens, and what happens? The sodium ions get transported inside. And such type of channeling is called as ligand gated channeling or ligand gated channels. So what is ligand here? It is the substance that which binds to the receptor or the protein here, binding site here. So sodium here is the bindings, is the substance that gets bound. Hence it is called as ligand. And this channel, the gate of this channel opens only when sodium ion, that is the ligand, binds, and hence we call them as ligand gated channels. And there are certain other channels which open only upon voltage difference. So let us say there are more positive charges outside and less positive charges inside. That means more negativity inside, less negativity outside. In that case, there is a difference in charges on either side. If there is a difference in charges on either side, there develops a voltage, and this channel opens only when there is a voltage difference on either side of the plasma membrane. In that case, we call these channels as voltage gated channels. So this is also having a gate extension. It can be like this or it can be like this. So it is closed, whatever it may be. In some books, this kind of a confirmation is given. In other book, this kind of confirmation is given. Whatever it may be, it is closed. So when this is open, either when a ligand binds, that is ligand gated channel, or whenever there is a voltage difference, that is voltage gated channel. Okay, and these are said to be most important with respect to understanding the nerve physiology. And coming to one more explanation I want to give you here. So there is a plasma membrane. Okay, this is a cell, extracellular fluid and intracellular fluid. This is cytoplasm with water and other ingredients, soluble substances and suspended substances. It is the cytoplasm, intracellular fluid, and there is an extracellular fluid that is nothing but the tissue fluid. So here, this membrane I have drawn here. So on one side there is extracellular fluid, then the other side there is intracellular fluid. Let us say there are sodium and chloride ions on either side. And here there are 90 sodium and chloride ions on one side and 60 sodium and chloride ions on the other side. Now what happens here? There is a voltage difference being established with respect to the number of positive and negative ions. In other words, we can say that here the more number of sodium ions are present on one side and less number of sodium ions are present on the other side. There is a concentration difference with respect to ions. So in any case, what happens? From higher concentration or higher area of what we call positive charges to the area where there are less number of positive charges, the ions start diffusing. So here diffusion takes place from high to low always. So what happens until, so about 10 have joined here, 70, it is now 70 and it is 80. Now another 5 moved and it has become 75 and 75. So when they reach equilibrium, When the number of ions is said to be same on both the sides, there is no more what we call diffusion taking place. Why? Because there is no difference in the voltage, neither in the concentration or the number of ions, nor with respect to what we call uh, Praveen Datta sir. Did you join? మమ్మల్ని మమ్మల్ని కనికరించండి సార్. అసలు పెద్దలు చెప్తాం అలా ఇల్లో ఇల్లో కొంతమంది అయినా సియు సెట్ సిఈటి క్వాలిఫై అయిపోయి సెంట్రల్ యూనివర్సిటీలో చదివితే నా కోరికలు తీర్చుకుంది. నీ చదువుకుందాం అనుకున్నా నాకు అవ్వలేదు. ఓన్లీ ఆంధ్ర యూనివర్సిటీ టార్గెట్ పెట్టుకుని అక్కడికి వెళ్ళిపోయాను. సో కొంతమంది నేను పంపించింది. ఓకే, థ్యాంక్ యూ. మీరు ఎగ్జిట్ అయితే నేను పాఠం చెప్పగలను, మీరు ఉంటే నేను చెప్పలేను. థ్యాంక్ యూ. Right. So sorry for the disturbance. So here, so when the equilibrium is reached, there is no change in the number or the voltage difference, so that there will not be any diffusion. And in such cases, no sodium and from this side or that side, okay, has what we call, in order to make it to equilibrium, no need for the sodium ions to move. And the same happens with respect to chloride ions. Okay, so as we have heard this, I want to ask you one thing. So now that there is 75 sodium ions on both the sides and 75 chloride ions on both the sides, that means it is in equilibrium. The membrane with respect to the concentration of the solution on both the sides is in equilibrium. Okay, my question to you is, in equilibrium, is there any movement of sodium ions and chloride ions or not? There is no movement. Any other answer? Any other answer? Is there any, at equilibrium, is there any movement of ions from one side to the other side or not? Hmm? No, you already have responded. I'm asking the other ones. Here is Vinay, Rishi, Bhargavi, Sanjay, Vasu. No. Bhargavi, equilibrium ఉన్నాయి కాబట్టి డిఫ్యూజ్ అవుతుంది. గుడ్. ఇంకా వైష్ణవి, పావని, Dimond, Deepak, Vasu. What about you? What do you say? No movement, Sanjay. Good. Any other response? If you don't want to speak, you can type like Sanjay. So what actually happens is, even at equilibrium, ions move. Why? Because they are in solution, isn't it? Here the water goes along with the substance that is dissolved in water also goes. Isn't it? Because these are diffusible ions. We call them as diffusible ions. So these diffusible ions diffuse. So let us say 5 ions have moved this side. Now what happened? 80 sodium and 70 sodium here. Now there is a voltage difference or there is an ionic difference, concentration difference, whatever it may be. Now what happens? Again 5 come back again to this side. So 5 have gone that side, 5 have gone this side, and the net movement is 0. So even at equilibrium, this you have to remember. Even at equilibrium, there is a movement of ions, but the net, what you have to see is, the net movement is 0. What is net? How many have gone that side? Have to be deducted from how many have gone this side, have come this side through the through the plasma membrane, which is in equilibrium. So when we minus these two, the result is 0, and this we call it as net movement. So net movement of ions during during at equilibrium is said to be 0. That means even at equilibrium there is movement of ions. Okay, is it clear? Good. Now we are coming to another situation. There is a membrane which is semi-permeable membrane, like the neuron nerve fiber. Okay, nerve fiber is said to be a semi-permeable membrane. On one side, there are sodium ions and chloride ions on the other side too. In addition to this, on one side, that is, let us say inside in the exoplasm, inside the cytoplasm, there are certain negatively charged ions which are called as, let us say, these are proteins. So proteins, we know they are said to be larger in molecular size and they cannot be transported. In case of neuron, there are other anions like phosphates, sulfates. These are also non-diffusible because there is no channel or no carrier that can transport a phosphate or a sulfate. So when such thing happens, we can call these as non-diffusible ions. So this is a non-diffusible ion and these are diffusible ions. Now what happened is there are about 90 sodium ions here, 90 chloride ions here, 90 sodium ions here, 30 chloride ions here, and 60 negatively charged protons here, proteins here. Okay, so totally how many? 90 and 90 here, positive and negative. 90 positive, 90 negative here. Do you say, is it in equilibrium or not? Why? There are 90 positive on that side, 90 positive on this side, 90 negative on this side, 90 negative on this side. With respect to voltage difference also it is same. What makes you say that they are not in equilibrium? So let me, there is a difference with respect to chloride ions. There is a difference with సో నెగటివ్ ఛార్జ్డ్ ప్రోటీన్స్ ఉన్నాయి కాబట్టి ఈక్విలిబ్రియంలో లేదు. ఇప్పుడు ఈక్విలిబ్రియం రావాలంటే ఏం జరగాలి? వాట్ హ్యాస్ టు హ్యాపెన్ ఇఫ్ ఇట్ హ్యాస్ టు రీచ్ ఈక్విలిబ్రియం నౌ? Which ion has to move? Chloride. How many? How many chloride ions have to move? So 30 chloride ions moved in. It means here it is 60 and here also 60. Now is it in equilibrium now? Hmm? Here 90 positive, 60 negative. Here 90 positive, 120 negative. Do you say it is in equilibrium now? So let me explain this. Let us say 90 sodium, 90 sodium, 90 chloride ions, 90 chloride ions. Is it in equilibrium or not? Is it? It is in equilibrium. So in equilibrium there are certain rules that we can see. What is that? The first rule is here the number of positive ions and negative ions on one side is equal to the number of positive ions and negative ions on the other side, isn't it? Here in this case. But here that is not possible here, isn't it? Then what the scientist says is this is not the case that is required here. They say that in equilibrium, the net movement of ions is zero. That is what we have already discussed. That net movement of ions is zero. This is the first rule. The second rule is the number of cations present on one side is equal to the number of anions present on the same side. Likewise, here also, number of cations present on one side is equal to the number of anions present on the same side. So, it is same on both the sides. And the third rule is, here these are diffusible ions. The product of the diffusible ions on one side is equal to the product of the diffusible ions on the other side. So when these three are said to be fulfilled, we say that this membrane is in equilibrium. Okay, did you note down? Should I explain it once again? Okay, then. Now what happens is, let me come back again to this particular thing. Here, there is a, what is this? Plasma membrane. 90 sodium ions on one side, 90 sodium ions on the other side. What about the other one? 90 chloride ions on one side, 30 chloride ions on the other side. And there are 60 protons on the other side. Okay. Now what happens is, because there is a difference with respect to chloride ions, the chloride ions tend to move. As someone said, about 30 chloride ions move from this side to that side, so that these are now 60 each. This is chloride, sorry. Okay, now these are 60 now. Now what happens here? The negativity increased 120, positivity is only 90. What we are saying, the number of positive ions on one side should be equal to the number of negative ions on the same side. Likewise, here also there are 90 positive ions and 60 negative ions. So to balance this, from this end, 30 sodium ions move to that end. Now what happens is, here there are 30, sorry, here there are 60. Now on the other side, how many now will be? Can anybody tell me? These are 120. Now the scientists say that this membrane is in equilibrium. This membrane is in equilibrium. But what is happening here? Here number of sodium ions is said to be different. Number of chloride ions is same, but there are more non-diffusible negatively charged ions on the other side. Still they say it is in equilibrium. So let us see these rules are applied or not. The first thing is, number of positive ions is equal to the number of negative ions on the same side. So is it true? There are 60 positive and 60 negative. Good. 120 positive and 120 negative. Good. That means the first rule is said to be applied. The second rule is said to be, here the product of the diffusible ions on one side is equal to the product of the diffusible ions on the other side. So what is the product of the diffusible ions? Here the diffusible ions are 60 into 60, that is 3600. Here the diffusible ions are 120, sorry, here it will be 30, 30, 30. So here it will be 120 into 30. It is again 3600. So what happened? The product of the diffusible ions on one side is equal. Then the third rule, what does the third rule say? Net movement of ions is zero. So when they are said to be it in this situation, if certain amount of sodium ions moved on that side, the negativity increases here. This negativity pulls back those number of positive ions that have moved on that side. Now what happens? The net moment is said to be zero. So in this place, we say this entire thing is said to be in equilibrium, and this equilibrium is called as Donan membrane equilibrium. That is what is responsible for creating the potential. In this case, here there are 60 positive sodium ions, 60 negative chloride ions, 120 positive sodium ions, 30 negative chloride ions, and 60 negatively charged non-diffusible ions. Okay, that means here the voltage difference is very much high. So there is a voltage difference on either side of this membrane, which is called as Donan membrane. The voltage difference seen on either side of this membrane is called as the potential. Because this voltage difference is seen on the membrane, we call it as a membrane potential. When this membrane, if it is not excited, that means you have not given any stimulus. If you uh, pinch me on my skin, you give stimulus. Without stimulus, what happens? There is nothing on the neuron here that are located in my skin. What happens? There is no excitability at all. That means this membrane is in resting state. So in the resting state, and the membrane is resting state, still too there is a voltage difference that is called as resting membrane potential. So Donan membrane equilibrium is responsible for the origin of resting membrane potential. So what is resting membrane potential? It is the voltage difference on either side of a plasma membrane that occurs due to Donan membrane equilibrium. What is Donan membrane equilibrium? How can we define it? This occurs whenever there is a non-diffusible ion. So it can be defined as like this. The influence of non-diffusible ion on the diffusibility of the diffusible ions resulting in equilibrium that generates a membrane potential is called as Donan membrane equilibrium. So once again I am telling you again this. The influence of why these are said to be unequally distributed, because there is a non-diffusible ion present on one side. So the influence of the non-diffusible ion on the diffusibility of the diffusible ions present on either side of a plasma membrane, which is semi-permeable, which is because it is not allowing this solute on either side of a membrane which is semi-permeable, leading to the development of a membrane potential, that is the voltage difference, is called as Donan membrane equilibrium. Now let us come back to the neuron. So this is a cyton and this is neuron. So I'm only taking this part for the explanation sake. So there is a neuron. Okay, I'm taking here the axon, axolemma here. So this refers to the plasma membrane, and this plasma membrane is a semi-permeable membrane. So we have a semi-permeable membrane. In the resting state, the ions are said to be having uh, a diffusion in this way. We measure ion concentration in milliequimolar concentrations. Okay, MEQ, equimolar concentrations. And what happens is, scientists say this is extracellular fluid and this is intracellular fluid. In the extracellular fluid, there are sodium ions, potassium ions, chloride ions and other ions, but we are with respect to nerve impulse, these three ions are more significant than the others. And inside also there are sodium ions, potassium ions, and chloride ions. But the distribution of these three ions is said to be different. For chloride ions, the equimolar concentration is 105, which is almost equal on both the sides. You can go. And coming to the potassium, on one side it is 10 MEq and inside it is 110. And coming to the sodium ions, inside it is 10 and the other side it is 120. Okay, so comparatively, negatively charged ions are distributed almost same, chloride ions, and with respect to positive, there are 130 outside, 120 inside. And in addition to this, the plasma membrane, the intracellular fluid is having certain non-diffusible ions. Just we have mentioned about non-membrane equilibrium, where the membrane equilibrium is said to be disturbed, is said to be reached equilibrium due to the presence of a non-diffusible ion, which influences the diffusibility of diffusible ions. So here there are certain non-diffusible ions like phosphates, sulfates, proteins, which are organic anions. We call them as organic anions. And all these organic anions are non-diffusible. That means they cannot pass through the plasma membrane. Plasma membrane is semi-permeable. It allows these three, but not these three. Okay, and there is a cell semi-permeable membrane which is not allowing these. So that what happens now? A membrane potential develops. So because of the presence of more negativity inside, more negativity inside. How this negativity is formed here? Because of the presence of more number of organic anions which are negatively charged. In addition to there are already chloride ions which are negatively charged, and the number of positive ions is less when compared to that of the exterior. Based on there occurs a voltage difference with positivity developing outside and negativity developing inside. So thus, because of the distribution of these ions, we say that there is positivity on the outside of the nerve cell membrane and negativity on the inside of the nerve cell membrane. Okay, and there is a difference in charge on either side. This difference in voltage or charge on either side results in a potential. Because it occurs on either side of a membrane, we call it as a membrane potential. Because this membrane is not yet excited, we call this as resting membrane potential. And some scientists wanted to measure this resting membrane potential. So he inserted an electrode inside and measured it. So when we insert an electrode inside, it obviously comes out with negative charge only. So the resting membrane potential is found to be having a negative charge, and this is minus 70 millivolts. So how the resting membrane potential develops? It develops due to the voltage difference or charge difference on either side of the plasma membrane, which occurs actually due to the unequal distribution of sodium, potassium, chloride, other organic anions, resulting in a positivity outside and a negativity inside. So the resting membrane should always have, remember this, positivity outside and negativity out inside. Then only it can be excited. So resting membrane with positivity outside and negativity inside is excitable. What happens if it is negative outside and positive inside? It is non-excitable. More negativity inside, less positivity outside, non-excitable. Whatever it case may be here, whenever there is positivity outside and negativity inside, it is excitable. So now we have seen what we call the development of resting membrane potential, which is minus 70 millivolts. Any doubts up to this point, please ask me. I'm going slow so that you can understand easily. No. Good then. Now what happens is, to this resting membrane, I am exciting by giving a threshold stimulus. What is threshold stimulus? The minimum stimulus that is required to excite a neuron or a muscle. So I have given a threshold stimulus. What kind of stimulus we can give? The stimulus can be chemical stimulus. So you put some what we call uh, amla inside your mouth. That is a chemical sense. The neurons there respond to the chemical. And if you use what we call temperature, the body can sense cold water and hot water. And it can be even physical stimuli. That means someone is hitting you. Okay. So these three different kinds of stimuli can excite a neuron. When it excites a neuron, what happens is there is origin of nerve impulse taking place. Okay, so this origin and propagation of nerve impulse and synaptic transmission I will explain you in tomorrow's class. So hope you understand up till this point. If any doubts, you please ask me. Otherwise, I'll stop here.